Everybody, this video is an overview of the Epithelial Tissues lecture by Dr. Chrisman. It's one of the first lectures you'll have where you probably haven't had the information before. And on top of that, it's a Chrisman lecture, and he's extremely detail-oriented. He wants you to learn how to be specific. You'll groan later. None of this is conceptually difficult, there's simply a lot of mental footwork. Be prepared to memorize pretty much everything. If you find yourself asking, do I need to know this? The answer is almost always yes. Also, this entire lecture is just a direct outline from this book, Chapter 5 of the 3rd edition, the Gartner and Hyatt Histology Textbook. And while Chrisman always gives you what you need to know in the notes, having a different means of integrating the information can be helpful for retention. There's also a few other figures and tables that he doesn't have in his notes. This is a quick video, so it can only focus on the big picture. Make sure you go over all the parts that I skipped, or even better, come to our tutor sessions where we can go into more detail. It's dangerous to go alone, so take these. There are a lot of structures with multiple names, and it can be tricky, but let's dig in. So what we're looking at here is a type of membrane specialization, and usually there's two different surfaces that we look at when we're talking about membrane specializations, the apical, or the top surface, and the basolateral, which is the sides and bottom. Now, on the apical surface, we have this structure called a microvilli, and we also have stereocilia, but stereocilia are actually microvilli, they're just a lot longer. And both of these are most notable by the fact that they're non-motile. They do not move on their own. So microvilli are very, very small, and they make up a brush border. So this figure should have about 6 just lined up in a row, one next to another, next to another. And in electron microscopy, they look very large, but when you're looking at a slide under a microscope, these are very, very small and hard to distinguish from one another. It just looks like a fuzzy border. Now, when we're talking about motile, or moving, self-moving membrane specializations, there are cilia, and then there are flagellum. And now, flagellum are cilium, but they have more mitochondria in them. And both are most noted by this inner structure here, this core called an axoneme. And they have a 9+2 structure in the middle here, where there's 9 pairs of microtubules and 2 in the middle, singules. And all of these have dynein arms attached to them. They're red here and they're black here. And these dynein arms are responsible for movement. Now, if you don't have these dynein arms, you will have a syndrome called Cartagener syndrome. And it's noted by, one, infertility, because the flagellum on the sperm will not be able to move, as well as constant lung infections, because these cilium are actually responsible for moving away mucus from your bronchioles and from your trachea. So not only can we talk about the classification of the structure of glands, we can also talk about the classification of how they secrete things. So there's actually 4 different types of secretion, but this figure only shows 3. The first kind we have here is called holocrine, and I'll just write this here. You'll have to forgive me for my handwriting. And holocrine is when a cell from the gland will fill with its contents, and it will just release from the rest of the gland and die, and then release its contents that way. And the cell will just sort of disintegrate after it's released. The second we have here is merocrine. And merocrine involves actually just releasing the contents of whatever you're trying to secrete just into the cell space here, and it'll just go into the rest of the body. And there's no involvement of cell death, and there's no loss of cytoplasm at all. And now this third form here is called apocrine. In apocrine, you will have the cell just blebbing off parts of its cytoplasm, which will carry the contents of whatever's being secreted. So there's just this pinching off. There's loss of cytoplasm, but the cells themselves don't die. And the fourth form is just cytocrine, which you will just find in reproduction is just the secretion of a living cell, so like an egg or sperm. Now that we've discussed epithelium and membrane a bit, we can talk about glands. Now, glands have a lot of hierarchy and structure, but we can walk you through it. First, glands can be exocrine or endocrine, which is just with or without ducts. And then they can be unicellular or multicellular. And in this figure here, we have multicellular exocrine glands. So this first gland here is a simple tubular gland. And this is just, tubular is just describing its shape, while simple is just describing its structure. And you can also have simple branched, which is just multiple branch points from this single stem. Now this is different, though, from compound, which is multiple branch points from multiple stems. And this caused a little bit of confusion last year, so it's important to try to make this distinction clear in your head. You can also just have sort of a different configuration of structure with this coiled tubular as well. Now, acinar, or also called alveolar, is just a bit more grape-shaped. It's a bit more bulbous in how it's presented. This figure does a bad job of showing it, but it's just sort of more rounded. And then you can have branched as well here, and then you can have compound as well, and then actually all of these can come together and you can have a compound tubuloacinar. So you can see how just a few terms can make describe a very complex structure. Now, this last figure is a bit overwhelming, but all we really need to think about are these highlighted terms here. So now that we've talked about how the glands can secrete things, we can talk about what they're secreting. So different sort of things our body secrete. There's mucus, there's serous secretions, which is like sweat, saliva, and tears, just much more watery. You can have a mixture of the two, and you can have sebum, which would be your sebaceous gland, so like oil on your skin. And then there's ceruminous secretion, which would be earwax. So each one of these is just describing, we have here, this is a serous cell in a serous gland, and this is a mix of the two of them, and this is a mucus cell. And you would likely find this somewhere in your mouth or perhaps nose. And this is just sort of showing you that these glands aren't distinct in the body, that they're all sort of mixed together, and you can have one spot with all these different sorts of ducts in them. So what we have here, it's a table of the classification of epithelia, and there's 4 main headings. There's the type, there's the shape, location in the body, and its function. You're going to want to know this table down pat. If you can, try and just write it out, make your own table, and just be able to fill it out from memory. For the sake of the video, though, we're going to be looking at the pictures so we can get these down. So there's 4 main types of epithelium. There's the simple, there's pseudostratified, there's stratified, and there's transitional. Now, simple is exactly what you'd expect. It's just a simple single layer of cells that are attached to this blue basement membrane here. And there's 3 subtypes for that. There's squamous, cuboidal, and columnar. Now, columnar sort of look like columns, cuboidal sort of look like cubes. Squamous actually has to do with scales, and so this is why you have sort of flat epithelium, squamous, flat, just get the association down. Now, pseudostratified, you have these staggered nuclei here, which make it look like it's multicellular, but actually all of these cells have a plasma membrane extension that connects to the basement membrane here, hence the name pseudostratified. Stratified are multilayered, have 2 main distinctions to get a hold of. First, both of these are squamous, but the difference is one is non-keratinized and one is keratinized. So you would say stratified, squamous, non-keratinized, stratified, squamous, keratinized. Now, keratinized is simply a dead layer of cells on the surface of the epithelium. Mostly you can think of your skin, just a dead layer of skin cells. That's the best way to think of it. Squamous non-keratinized, that's what you're going to find on wet inner tissues like your esophagus. Now, the other distinction, of course, are still, again, cuboidal and columnar, but now they're stratified, so you just have a multi-layer of them. Finally, there's transitional, which Dr. Chrisman has this as part of the stratified, which they're multi-layered, but their shape depends on whether or not the tissue is distended or relaxed. And transitional is also known as uroepithelium for the urinary tract. And so when the urinary tract is relaxed, they sort of look more like the stratified cuboidal over here, but when it's distended, it looks a bit more like the stratified squamous, depending on its shape. So this is a figure of the basolateral membrane specializations. In each cell there's a zone, and within each zone there are specific structures that are found within that zone. So for the zonia occludens, you'll find tight junctions. And a little factoid about this is that occlude is actually the opposite of include, and that's exactly what tight junctions do, would be the opposite of including things in the cell, so they keep things out of the cell. Now, the zonal adherens has the adhesion belt, the macula adherens has desmosomes, and Chrisman often describes these as spot welds. And the important thing to know about desmosomes is their associated autoimmune condition called pemphigus vulgaris, which sort of sounds like a Harry Potter curse, I'll give you that. But what happens is that antibodies will actually attack these proteins called desmogleins, which make up the desmosomes here. And the result is very awful blisters, and they actually result in life-threatening infections because of how bad these blisters are. And finally, you have gap junctions, which Chrisman notes that there also can be called nexus, but you'll almost always hear them as called as gap junctions. And this is the lateral side, and here on the basal side, there are some membrane enfoldings, but mostly you'll want to know about hemidesmosomes here. And the protein that's involved with hemidesmosomes is called integrins, and these proteins help to connect cells to the basal lamina. Well guys, that's all I'm going to be covering for this video overview. I hope it helped. My name is John Lukoski, and I wish you all way more than luck.